I'm going to tell you a little bit about a evaluation of a proteomics approach for detecting rice allergens. My colleague Jean-Baptiste Rasclé has been the coordinator for this project, but he couldn't be here, and so I agreed to come and give you an update. I will start with a little background on food allergy, rice allergy, and explain the rationale behind the project, and then tell you where we are at present. So fortunately, rice allergy is pretty rare in most areas of the world, except in areas of high rice consumption, and particularly in populations that are prone to developing allergic disease. So for example, the prevalence in Japanese atopic patients for rice allergy is about 10%, and that figure comes from a couple of older studies from the 90s. The frequency of rice allergy is much lower in the US and Europe, and tends to be higher in adults than children. Symptoms include the usual IgE-mediated unpleasantries, but also rice allergy has been associated with atopic dermatitis, asthma in some rare cases, and in some severe cases, anaphylaxis. In terms of the allergens themselves, there are several that are fairly well characterized. There's an important group of proteins in the 14 to 16 kilodalton range that are in the alpha amylase trypsin inhibitor family, and these tend to be immunologically cross-reactive with each other, but also homologous with other proteins in the same family of other cereals, and are probably responsible for some of the cross-reactivity between the cereals. There's a 33 kilodalton glyoxylase that's been identified. And then there is a series of globulin-like proteins that have recently been identified as being immunologically reactive. And then there's a pollen allergen, too, that plays a role in respiratory allergy. But these guys that are listed in red here have been the focus of the project. So the project is focused on evaluating the 2D Dodge method for detecting rice allergens. And 2D Dodge is two-dimensional difference gel electrophoresis. It's an advanced version of the two-dimensional SDS page. And the ultimate goal here is to understand the variability that exists among plant proteomes, because without that information, it makes it very difficult for us to interpret changes in expression levels of proteins when the plants are genetically modified in terms of biological relevance and safety. So there was a paper from Dr. Reiko Teshima and her group that reported that this method was useful for analyzing the natural variation in rice varieties, and they were able to compare the level expression for allergens as well. And so the project goal was to assess the levels of expression of some of these rice allergens in 5 independent laboratories to determine how reproducible this method is. So the project outline, we're calling this phase 2, phase 1 being the 2010 paper from Dr. Teshima, and it's focused on 3 rice allergen families, the 52 kilodalton globulin, the 33 kilodalton glyoxylase, and then that family of alpha amylase trypsin inhibitor members. There are 4 rice cultivars involved. None of them are transgenic. And then there are 5 laboratories. Several of them are academic. There are 3 in the United States, University of New York at Buffalo, Donald Danforth Plant Science Center, and University of Nevada at Reno. And then in Japan, there's a National Institute of Health Sciences and National Food Research Institute. Dr. Teshima is the project lead on this. And then, as I mentioned earlier, the project coordinator is my colleague, Jean-Baptiste Rasclé. So in terms of the procedure, this technique is very similar to the original two-dimensional electrophoresis. They share these first 2 steps in that they both require protein extraction and then protein cleanup. And then the processes diverge here because the 2D Dodge uses fluorescent dyes. And so the imaging is a little bit different as well. But the procedure is based on that 2010 paper from Toshima et al. So it all started with rice, 100 grains for each rice variety, and these came from the World Rice Collection. They were all processed at NIHS in Japan, and then put on dry ice and shipped to the labs that were participating in the ring trial. And then each lab did the protein extraction and the protein labeling. So the proteins are labeled with 2 different fluorescent dyes. And then each sample, a little bit of each sample goes into a reference pool that's labeled with a 3rd dye. Okay. And then they're run out on gels. And so the study design is a quadruplet hit method. And so what happens is the reference pool goes onto every single gel, and it serves as an internal reference or internal control for staining. Then each variety is incubated with 1 dye or another dye. And so this helps us account for labeling bias. So everything gets run out in duplicate, and the gels look something like this. So the proteins are separated based on their isoelectric point and then their molecular weight. And so this is the gel from the original paper from 2010. And you can see here that the allergens are being detected in these areas. So these are spot picked, and then they're sent for mass spec to identify them. And here are the results from the 5 labs. Up top are the results for the 52 kilodalton globulin. So here's the results from each lab. And they're fairly consistent in the number of spots they're able to detect between labs. The same is true for the 33 kilodalton glyoxylase. And so you can see that they're getting basically the same number of spots. And then when we get to the alpha amylase trypsin inhibitor family, things sort of fall apart a little bit. We're getting 5 spots over here and something like 20 spots in here. So there were some differences there. We don't have to strain our eyes staring at this, though. We've got some nice graphs. So each graph represents a spot that was detected in common by each laboratory, okay? And each laboratory gets a different color. The legend is here. The y-axis is the normalized value, so that's the value of the fluorescence intensity ratio to the internal standard. And the rice varieties are here on the bottom. So for each rice variety, there's a spot. And the way that these lines track together is an indicator of how reproducible the technique is. So when labs are getting the same results, then the differences in the spot between each variety will be fairly consistent across labs. And we're seeing that here for the globulins. That's also true for the glyoxylase. But the difference here is that the lines are all relatively flat. So what we're seeing is not a lot of difference between the varieties in terms of expression of this glyoxylase. And then, of course, this alpha amylase trypsin inhibitor family, you can see there's a lot of variability, both in terms of expression and in terms of the results that the different labs are getting. So the conclusions from this were that the 5 spots that were detected in common for the globulin showed consistent differences in their expression levels in all labs. The 33 kilodalton glyoxylase was detected by all labs and did not show any significant differential expression between the different varieties of rice. And then the detection of this family of 14 to 16 kilodalton proteins was a bit more variable. Some labs detected 6 spots, some labs detected 20 spots. But when focusing on the 5 commonly detected spots, the data were not consistent enough to reach conclusions about how they were differentially expressed amongst the cultivars. So this is what we think is happening based on some pilot experiments. it seems that there are potentially issues with protein extraction and cleanup methods. That is that, technicians are reporting that they're getting different size pellets when they're really starting with all the same starting material. And so the thinking now is that the next step is to optimize these protein extraction methods and see if we can get a little more reproducibility. So we want to acknowledge the project participants and the PATC for their support, so thank you to them. I have not been directly involved in this project, so I will try to answer your questions as best you have if you have them, but in any case, thanks for your attention.